Conceptually, when we approach tree breeding, it's important to keep in mind that we're dealing almost always, at least in forest trees, with polygenic traits, traits that have a continuous distribution. So for instance, if we have a particular trait here on the x-axis with values of that trait from low to high, that might be height or biomass at a particular age. It might be some wood property. It might be something to do with disease resistance. That there's a continuous distribution of those values in the population, not always normally distributed, but in this case, maybe normally distributed. So there's some mean value of that trait in the population. And the idea with tree breeding is that we're selecting individuals that are out on one end of the distribution that are better for that particular trait, and then using them as parents in our population. And that points out another thing with tree breeding is that usually we are using or evaluating parents and whether their offspring or progeny perform well. So breeding is done on parents, but we're generally evaluating the performance or the traits in the offspring or progeny. And that, I think, will become more apparent as I describe the structure of tree breeding programs, and you'll see the role that parents and progeny play in that whole process. So what kind of traits are generally of interest? Probably the most commonly used trait, especially in commercial tree breeding programs, is growth rate. This is a 2-year-old loblolly pine in a genetic trial, and it's pretty big. Now, of course, that's its phenotype, so we don't know whether that tree is genetically superior or not, but generally, growth traits are the most commonly used traits that are selected for tree improvement. Disease resistance is probably the second most common trait that is associated with tree breeding programs. This is an example, sort of an extreme example of a southern pine tree with fusiform rust disease. Fusiform rust is the most damaging disease agent in southern pines, economically at least, and it has been extensively improved in populations of loblolly and slash pine. So incorporating resistance to disease is probably the second most common trait. White pine blister rust might be another example of an important disease that is being incorporated into tree breeding programs. Characteristics, there are other properties that have been incorporated to variable extents in breeding programs. So form of the stem, so things like the presence or absence of forks, stem straightness, those types of things affect the value of wood that's derived from trees, and to some extent some of those have been incorporated into selection indices in breeding programs. Wood properties like specific gravity can be an important trait in improvement programs. Some eucalyptus breeding programs have selected either parents or clones for wood chemical traits. Remember a while back when we talked about wood properties, we talked about S and G lignin and their relative ratios, that has been used as a selection trait in at least eucalyptus I'm aware of. Aesthetics properties are a potential trait in breeding programs. This is a beautiful red maple tree in a yard. So this is likely a variety that was purchased from a commercial nursery, and that variety was likely selected for fall color. Things like leaf shape, bark attributes. So there are some trees that ornamentally their bark is a big characteristic, so they might be selected for bark. Size. So, I've mentioned growth rate, but certainly some, especially ornamental varieties, might be dwarf varieties. They've been selected to be small because that's sometimes desirable in ornamental situations. So any trait that is inherited, in other words, any trait that has some non-zero heritability, could potentially be incorporated in a breeding program. Now, for some of these traits, like leaf color and especially ornamental traits, those aren't always used in a formal quantitative genetics breeding type program like I'm going to describe. Sometimes individuals that have those traits are selected, and then they might be vegetatively propagated. So sometimes the selection is less quantitative and more informal or qualitative. But at any rate, those traits are still heritable in that they can be reproduced either by seed or, in the case of clonal reproduction or vegetative reproduction, they're just reproduced clonally. But at any rate, the list of traits is quite long that can be improved for. I'm going to walk you through the steps in a tree improvement program, and I'll sort of walk you through this as if we're dealing with an undomesticated wild species and we're taking the first steps in a tree improvement program. These first steps were taken, for instance, in loblolly pine back in the 1950s. So this first step was taken in the 1950s. And it has continued for multiple generations since then. So the first step is to go into wild populations and identify phenotypically superior trees. So walking into a natural stand, looking at the trees and saying, that is a good looking tree that has the traits that are desirable. So for instance, this is a loblolly pine tree growing in the Hatchet Creek bottom near Austin Carey Forest, has a big red mark on it. It's growing wild. This is a wild loblolly pine tree, and it was a phenotypic selection in the tree breeding program here at the University of Florida. It's big, it's straight, it's relatively free of disease, good stem form. It's phenotypically superior. So that's an example of a wild selection. It's growing out there in the wild. So cuttings are taken from that tree. Obviously, it's a bit of a challenge with a big tree like that, and usually those cuttings are taken with a shotgun. They, shoot at the branches of those trees and with the shotgun, and branches fall down. And then they take those cuttings and graft them onto rootstock in a seed orchard. So you establish a location, a central location, and you've planted seedlings in that location. They're just growing as seedlings. And after a year or two, you come along with those cuttings from the phenotypically superior wild phenotypes, and you graft those cuttings. Those cuttings are called scion. Okay, scion. Those are grafted onto that rootstock in the seed orchard. And that cutting, if it's grafted properly, will grow and become established, and you cut away all the branches from the rootstock so that eventually all you have above the graft is material from that wild selection. And you make multiple grafts from one selection. So now you have multiple copies of that genotype in the seed orchard. So this is an example, this is an aerial view of a seed orchard. These can be very large, and there might be many, many copies of that wild selection genotype in a seed orchard. And these seed orchards are managed to maximize the production of seed. The trees are far apart, they're mowed and fertilized, so the crowns get really big and they produce lots of seed, because that's the purpose of a seed orchard. So once you have that seed orchard, you can use seed from that seed orchard to quickly establish new commercial plantations from cones or seed collected from that seed orchard. So when you're thinking about this, these seed orchards are phenotypically superior. We don't know that they're genetically superior yet because, again, phenotype is a mixture of genetics and environment. And right now, all the selections we have in that orchard, we know they're phenotypically superior. They looked good in the woods, and now we're breeding them with each other because these are all pollinating each other in the seed orchard. But we don't know how much of that is genetics and environment. But at any rate, we can use those to quickly establish commercial plantations. Just this first step, these commercial plantations established from that phenotypically superior subset of the wild population might already have a 5 to 10% improvement over wild populations. So basically that's the improvement that you get just because some of that phenotypic superiority that you brought into the seed orchard, some of that is genetic. And that is carried through into these offspring, these progeny. So just that first step will give you a 5 to 10% improvement over wild populations. But at the same time you're establishing these new plantations, you also establish a number of common garden studies across the area where you would like to reforest with these improved populations. These common garden studies, where you're planting basically offspring from many different parents in the seed orchard, you plant them, all those offspring from many parents. So this might be offspring from parent 1, offspring from parent 2, offspring from parent 3, and you have those offspring from the same parents in many different locations or in multiple different locations. Okay? So each of those is a common garden study. And they're the progeny. We're not planting genetically identical material to what's in the seed orchard. We're gathering seed from there and planting the progeny. So what we're going to do is use information from these progeny tests. That's what these common garden studies are called. They're progeny tests. We're using information from those progeny tests to understand the value of the parents. Do the parents in the seed orchard produce progeny that perform better than average? And this is an example of a progeny test. You measure those progeny and say, is this a big tree? Are there other traits that we're interested in? And we measure those in the progeny in these different progeny tests. And this is where quantitative genetics comes in. Quantitative genetics are used to analyze the data from the progeny tests to basically separate out the effects of environment from genetics. So keep in mind, what we have here is, because we have these common garden studies, we have progeny from many parents growing in one location, and in that location, environment is relatively constant. So this might be environment 1. But we have other progeny tests growing in different environments. So we have tests of these offspring in many different environments, and that enables us to use quantitative genetics to separate out the effects of those different environments from the genetic effects on the traits of interest, whether that's growth, disease resistance, whatever. Okay, so now that those quantitative genetics measurements, assessments, those then tell us, those allow us to rank the genetic worth of the parents in the seed orchard. And they allow us to then determine, oh, okay, parent A, B, and C clearly produce offspring that are better than average, whereas parents D and E don't. It's likely then that parent D and E, while they were phenotypically superior in the woods when we selected them, most of that phenotypic superiority was due to environment. But those progeny tests and the quantitative genetic analysis allowed us to tell that they were not genetically superior. Once we know that, we can go back to the seed orchard and cut down the parents, remove the parents that are not genetically superior. That process of cutting down the genetically inferior parents is called roguing the seed orchard. We're getting rid of the rogues. We're roguing the seed orchard. That whole process, from selecting wild phenotypes all the way around through the cycle of testing and roguing, that is one generation. That's one full generation of improvement. The first generation of improvement in, for instance, loblolly pine took around 25 years. And if you think about it, you can think about biologically why this takes a while. If you think about how long each step takes, okay, here where we graft scion into the seed orchard, those grafted cuttings don't produce seed until that cutting becomes mature. So it might take 10 or 12 years to produce seed. So maybe this step is 10 to 12 years to produce seed. Okay, now you've got seed and you establish these commercial plantations and you establish progeny tests. Well, those progeny tests have to grow some period of time. Well, you have to establish them, and they have to grow some period of time until you can evaluate them. So this step may be another 10 to 15 years, because also you need to have enough seed from the seed orchards to even produce the progeny tests. So that whole step can be 10 to 15 years. You've got to analyze the data and then rogue it. So that all that together, you can see you're looking at 20 to 30 years for one generation. So this is a long-term process for forest trees just because of their biology. They're long-lived species. They don't reproductively mature for quite some time, and that causes this to be a slow process. So once I meant to mention, once that roguing occurs, once you remove inferior individuals from the population, that prevents pollen contamination of the material in the seed orchard because you've removed that inferior pollen from that seed orchard. And at that stage, you might have, in the neighborhood of a 10 to 20% improvement relative to wild or unimproved. And I'm speaking vaguely. Most of the time that improvement is in growth rate, but it could be in any of these traits. So at any rate, um, that's sort of what you can expect from one generation. In southern pines, we are now in or completing our third generation of tree improvement. So if you compare that to, for instance, other commercial crops like corn or wheat, those species have been through thousands of generations of improvement. Obviously, most of that was not formal improvement. People just collected seed from individuals that bore a lot of seed, for instance, and kept some of that as seed corn or wheat for regenerating the next year. But that resulted in genetic selection, and that's been done since the beginning of agriculture. So the commercial food species we have now really don't resemble their wild progenitors. Modern corn does not resemble its wild progenitor. It looks very different. In contrast, we're only in the third generation of breeding in southern pines, and if you look at a genetically improved southern pine, third generation, and compare it to a wild tree, you can't really tell the difference just from looking at it. So they're still essentially wild. That doesn't mean they're not different. They are different. They grow a little faster. They're more disease resistant. But I'm just trying to emphasize the difference in scale and time that has occurred in, for instance, food crops versus tree crops. Big difference. So multiple generations. So once we go through that first generation, I sort of showed you what happens in that first generation. You don't throw it all away, but you might, for instance, infuse some new selections from the wild into the program. You might make some new crosses, or you might do some full sib crosses, and in that process establish new seed orchards and new plantations and do additional breeding and testing. So that process just continues iteratively, generation to generation. And the improvements that you see, this is examples of improvement. So the y-axis is harvest volume compared to wild. So these are percent increased harvest volume relative to wild populations. The blue is basically individual trees. How much faster do they grow? And the orange takes into the account the fact that those improved trees are also rust resistant, so there's better survival in plantations. So that's sort of the fully realized effects of genetic improvement. And what you can see is that this 1.0 unrogued, that's that first step I told you about, where you've established that seed orchard with phenotypically superior material, but you haven't gone through the testing cycle yet. And what you can see is that there was, about a 15% improvement just from those wild selections. But then they went through and did testing and rogued the seed orchard, and that gave them maybe another 3 to 5% improvement by getting rid of those inferior individuals. And then they infused new material into the population, establish more plantations with that, get a little bit of improvement, do another cycle of testing and rogue the seed orchard, and they are now at that second generation, 32% improvement over wild. And you can see by the end of the third, well, this actually isn't the end of the third, this is that third generation that hasn't been rogued. It's over a 50% increase over wild populations in harvest volume. That's a big improvement just through selection and testing. So this is an example of the magnitudes of improvement that you see. And so this is basically over about a 50-year period. If you took wild seed and planted it, and next to it you planted a stand of third generation unrogued material, that third generation material would produce 50% more volume at rotation age than that unimproved material. That's pretty impressive. So that's sort of the scale of improvements that you see with modern tree improvement.